Yeah, it's my pleasure to be here for Pippa's defense to see the culmination of the work that she's done in our lab with many members of our lab, in fact, and outside our lab. We first met Pippa in 2022, of all things. She approached our previous head of lab, Dr. Jerry Bartholomew, who's since retired. You outlasted her, Pippa, but not your fault. And really, anyone who knows Pippa will know the enthusiasm she brings to the room, and that was true of her experience in our lab too. She also excelled at working with a range of people, both from sort of student peers, grad students, and both myself, other academics, and even outside our campus, Dr. Dorothy Huchon, who's one of the authors of a core study that's peripheral to Pippa's work. Dr. Huchon is also in the room on the Zoom here and mentored Pippa with a lot of the bioinformatics components that you will see her present. So with that, Pippa brought this ability really to work well in many teams and is in that sense a role model for a lot of students coming into the lab. Her project was not a simple project. It was challenging. As you will see, she encountered many, let's try this, that doesn't work, let's try something else. But I think really that type of project is what the BRR program welcomes, in that it's not just a simple, here, follow this protocol that someone's developed, get a result, write a paper. No, it's more the real world of science, throwing you in there. Here's where we want to head. Here's the intellectual backing, but you have to come up with the hard work. And so I'm happy to turn this over to Pippa now and let her present this work that she's done in our lab over the last 3.5 years. And take it away, Pippa.
Awesome. All right. Let me get that changed. Sweet. Thank you, Stephen. Those are really kind words, and I really appreciate that. But today I want to talk to you all about my project. The powerhouse of the cell has a blackout, searching the genome of Myxozoan parasites for mitochondria. My full name is Philippa. If you've seen that, I'm not just hiding behind an alias. And I'm part of the Parasite Host Interaction Lab with the College of Ag and also the College of Science. All right. So I just want to start off with some acknowledgments. I really want to thank everybody in my committee. In multiple ways, probably known and unknown, you guys have pushed me, encouraged me to grow, and really made me a student of life and a student of school. And I really just thank you all. I also want to thank everybody in the lab. You guys have been witness to a lot of my groans, grumbles, and gripes with the issues that have gone on. But you also have pushed me and shown me that I can be a lot smarter and keep questioning and keep criticizing. And I really thank you guys so much. As well as Kristen, she's not here, but I just really want to say she was so helpful with one of the big challenges that I'll talk about later in the project. And it's just amazing to work with her and hear her stories. And then lastly, thank you, Wanda and Gail. You guys have been amazing and pushing me to take classes I never thought I could even dream of taking. One of which was Dr. Weisberg's amazing class. I just want to thank you all for showing up and for being here, because it's much better than presenting to my stuffed moose. All right. So my personal background, while also a scientist, I am a rat guide. And this is a hobby, it's a leisure activity, and it's a job. But more importantly, it's how I can communicate science. And I'm really passionate about communicating science to anybody in any walk of life. So when people are stuck in my boat, they're locked in there, they're in jail for like 2.5 hours with me. And that's where I kind of inundate them with all the information I can about the river, and let them know the problems, but also the really cool facts that are, right under their feet or their butt. It's in the boat. All right. Okay. So for the project overview today, I'm going to introduce the system that we're talking about. Then I'm going to go into our main parasite of study, which is Myxozoan insidiosus. From there, we'll talk about all the challenges and successes that we had. Then we'll transfer into Myxozoan cerebralis, which I'll explain why a little bit later, and then into the discussion where we go from there. Okay, so initially, the endosymbiote theory, it explains the acquisition of mitochondria. Mitochondria, many people know, are the powerhouse of the cell, but eukaryotes or animals, we didn't used to have them. Millions of years ago, an ancient bacteria, which was able to produce its own energy, merged with an ancient archaeon, And that merging was so beneficial that those cells continued down that line, possessing the mitochondria, and ultimately created eukaryotes, which is you, me, trees, squirrels, rats, animals, basically. And with that, there's theory upon theory upon theory and theory within the endosymbiote theory itself. But the main takeaway is that after this merging, there's two distinct genomes. There's a nuclear genome and there's a mitochondrial genome. And that's really the point that I'd like to make right now. All right. Since then, we have made some observations that there have been mitochondrial gene losses. And so the canonical mitochondrial gene, or the mitochondria, the textbook mitochondria, it can do oxidative phosphorylation. It can create ATP. It can create energy. And it's a standalone organelle. However, through successive losses in the mitochondrial genome and the losses of those genes, the function of the mitochondria begins to erode. And that turns into anaerobic mitochondria. So generally in a hypoxic environment, you don't have the oxygen that mitochondria need. So you kind of start to lose those genes that need the oxygen. Ultimately, these eventually form a mitochondria-related organelle. And sorry, the mitochondria-related organelle is the range of these, but it can eventually form a myxosome or completely disappear in the case of one such species. Okay. Certain mitochondrial genes are more conserved across species. So this is a figure pulled from Glistad and Johnston, and it's a phylogenetic tree that shows the organization of species and the mitochondrial losses they possess. So we have, see up here, these are all clades within the tree of life. We've got metazoa, fungi, ciliates, apicomplexans, and chlorophytes. Humans are right about here within metazoa. Down here on the lower x-axis, this is detailing the presence or absence of canonical mitochondrial genes. So we've got C1, C2, C3, C4, and then some other ones that we can talk about later. Zooming in to metazoa, this is the same figure, but just turned sideways. Again, humans are right here. We want to look specifically at Hennegaia salmonicola. This is great because it's a Myxozoan, and this is what our lab studies. But Hennegaia salmonicola, it has some of the canonical genes, but not all of them. Importantly, Hennegaia salmonicola, it exhibits a characteristic called tissue tropism. So these here are salmon fillets, and the tissue tropism, it's where the parasite chooses to reside for some reason, but it's where it lives within an organism. So as I mentioned, it's in the muscle tissue, and it forms these kind of white, fat-looking cysts. And this is a zoomed-in version of that. Right. Importantly, your environment drives your development. So if you're familiar with Darwin's finches, their environment drove their beak size. So we want to see, is the environment that they live in, the muscle tissue, a driving factor for their losses in the mitochondrial genome? All right. And now, all the way into our first parasite of interest is Myxozoan insidiosus. And this is the one that we're going to talk about and our parasite of interest. It is also a parasite of muscle tissue, but it has an uncharacterized genome. You can see in the central image, this is one of those cysts, zoomed in very far. And then these are the muscle tissue surrounding with the striations. This image on the left shows the spore in its unfired state, and this valve shell around the parasite, and then image on the right is its fired state. Right. Importantly, with M. insidiosus, it doesn't necessarily cause mortality within salmon. It's found within Chinook and steelhead, but like I said, it doesn't cause inherent, death within a fish. That being said, it does cause environmental and economic loss. So imagine you're a fisherman, you're out, you make your livelihood off of the quality of the fish that you can bring in. And you cut it open and you see those white cysts in the fish muscle. That's not going to really sell. So you either throw it out or you stop fishing. So that loss is the environmental and economic loss that we see with M. insidiosus. All right. All this culminates in our main driving question. Do parasites residing in the muscle tissue have all of their expected mitochondrial genes? I mean, really, is that ecological niche that they live in a driving factor of their genome reduction? To answer that main guiding question, we outlined smaller guiding questions. We're doing this in stair steps. First, does M-insidiosis have visible mitochondria? That's our big initial question to ask. And second, if they do have mitochondria that we can see, do they have all the expected genes within their mitochondrial genome? So to answer our first question, that occurs through fluorescent microscopy. To answer our second question, that occurs through genetic sequencing and assembly. To get right on into it, the fluorescent microscopy, it revealed visible mitochondria. Yay! We've answered a question. You can see here in these images, this one not so much, they're a little bit faded, but this one really well. These small sprinkle-like structures that we're assuming are the mitochondria. Our positive controls to show that the staining actually worked was the fish or the spore nuclei, as well as fish nuclei that aren't shown in the images. Right. So, does M. insidiosus have visible mitochondria? Yes. First question, done. To answer our second one, it's a little bit more involved, but we wanted to do genome extractions to collect high molecular weight DNA, and that was for the sequencing and assembly steps. That starts with extracting and isolating the spores. Then we lyse those spores to try and release that DNA into solution, and then we isolate that DNA so we can send it off for sequencing. The reason we want high molecular weight DNA is for later steps. High molecular weight DNA is DNA in super long strands. And if you imagine putting together a puzzle that is 50 pieces large versus a puzzle the same size that's 1,000 pieces, that 50-piece puzzle is going to be a lot easier. So that's why we want super long strands of DNA to put together that genome as easily as possible. All right. To do this, we excised fish muscle tissue, and that was to get the spores. So we took our salmon fries. These were euthanized at the Aquatic Animal Health Lab out in Eastern Corvallis. We dissected vertically along the gill line, horizontally along the spine, and then pulled back the tissue to expose the muscle along the spine. We then took all of that tissue, as much as we could get, and put it into a falcon tube and called it the spore slurry. Notably, we really wanted to avoid getting tissue very close to the spine or any cartilage, because there are other parasites that we don't want to sequence in those tissues. All right. With the tissue in the spore slurry, we let that settle overnight into this pellet here. There we go. And then we took it through Percol gradients. Percol gradients were important to use because they helped us reduce the fish contamination and moderately reduce the bacterial contamination. What the Percol is it's a high density fluid that we were able to place the spore slurry on top of right there. And then we spun it down, and with the centrifugal force, those spores settled all the way to the bottom, and we were able to collect that pellet. We ran it through a second Percol gradient to refine it even more. And ultimately, right there in that super faint line, that's where our spores pelleted out. Let's see. With this, we also did spore checks because we can assume we're reducing the contamination, but we want to verify. So as you can see here, this is that spore slurry. It's got a lot of gunk in there. Loving gunk. There's maybe 1 or 2 parasites that we 1 and 2 spores of M-insidiosis, but largely it's a lot of fish tissue and a lot of things that we don't want to waste time and money sequencing. At the end of that, that's our final pellet. Y'all can see the clear difference between those two. We've got a lot of spores in this one and maybe some bacteria with those little black dots. We've reduced the fish tissue contamination, which was our goal with that. All right. Once we took that tissue, the isolated spore pellet, we wanted to extract the genome from it. And this is kind of where a lot of the challenges occurred. But the extraction protocols we used had varying degrees of violence associated with them. And that's just a colloquial way of saying we'd beat the spore in different ways to try and crack open that shell that I pointed out earlier. So initially, we had tried liquid nitrogen grinding with a frozen mortar and pestle. We then tried an extended vortex with garnet beads. And in our beautiful jerry rig system, we stuck a vortex in the incubator and taped it closed and tried that method. All these ultimately, we also did spore checks during that time to make sure we were actually cracking open that spore and releasing the DNA into solution. And as you can see in this first image, pre-genome extraction, this is a methylene blue stained sample. So with that, the spores, they're actively keeping that stain out of them. That's telling us they're alive, they're synthesizing, they're keeping that stain out. They still have membrane integrity. There you go. With our second image, while this isn't methylene blue stain, you can see in these two locations, the spores look 2D. They've lost their internal components, and so we're making the assumption that the DNA is now in solution. It's all around in there, and there are some spore shells, and that's what we wanted to see. All right. But, well, after that, we ran it through a gel, and we got negative results from the gel. This was kind of a rough and tumble way to see if there was DNA presence, but it's not finite or super assured. So we went to a qubit assay to try and get the actual DNA concentration in solution. And with our genome extraction results, we got nada. In reality, we did actually get a quantity of DNA back within the samples, but it was 1/100th of a nanogram per microliter, which is far below the actual sequenceable level for PacBio, the sequencing program we wanted to use initially. So it's basically nothing. It's kind of a problem. We can't answer our question. But going back to our guiding questions, we did find out that it has visible mitochondria. In terms of if those mitochondria have all the expected genes, we don't know. Basically, investigating the M. insidiosus genome, it requires deeper analysis and much more refined extraction protocols, and that they were just beyond the scope of this project. We set out with the goal to find what was in the genome, not to find the genome. So we're not left with nothing. We're scientists. Everyone in this room has had problems in life. We go back to the drawing board. And, we can either keep hammering away or we can change our subject and keep going. So that's what we asked ourselves. Where do we go from here? We either stay with M. insidiosus or we hardcore crank to something new. That's what we did. We asked ourselves, what species do we know have undergone mitochondrial losses within Myxozoa? That's what our lab's did. We have Hennegaia salmonicola. I mentioned that earlier. But also, I didn't tell you about, there's Myxozoa squamalis. And Myxozoa squamalis has also undergone mitochondrial losses. So once we know that, we asked ourselves, what's a closely related species to M. squamalis that has available genetic data? And that's Myxozoa cerebralis, or MC. Myxozoa cerebralis is the causative agent of Whirling disease, and it's a harmful pathogen that affects fish aquaculture worldwide. Specifically, and it affects the lower Deschutes River where I guide, so it's important to me as well. So it shares a similar life cycle to other Myxozoans in that it has the two-host, two-stage life cycle. And the data we have from this is right here. We were able to find transcriptome and genomic data from the triactinomyxons, or TAMs, and these are the parasite life stage that infects the worm host. All right, and we also got kind of lucky because there's a very recent partial genome that was sequenced that we were able to look at. So we asked ourselves then, does the M. cerebralis transcriptome or genome contain all the expected mitochondrial genes? And to figure that out, we had 3 steps. We got the gene sequences, made sure they were trimmed and cleaned. We searched for genes within those sequences, and then we verified that any positives we got back were not contamination. We searched for 5 genes in the M. cerebralis transcriptome using sequences from representative species. So from Myxozoa squamalis, we used the NAD1 and NAD5 genes. And then from these 3 species, Myxozoa shantigensis, Myxozoa hongoensis, and Tilonellus katawi, we used the COX1, COX2, and COB genes. These were our references that we used. For the specific workflow, I'm going to go through this quite slowly because it is pretty detailed. We took those reference sequences, and these became our query. So we queried the Myxozoa cerebralis transcriptome, or dictionary. That dictionary had a number of sequences, but we didn't know what they were. So any matches that came back between these two, we found. And that was either positive or negative hit or unknown. With those positive hits, that's great. That means we're getting data back. But we got to make sure it's true data. It's not some contaminating sequence. So we collected all of those positives, pulled them into a single database, and then we searched that database against NCBI, which is a genetic sequence repository on the internet. That actually helped us a lot because it showed that we did have contaminating sequences. Any NAD5 and NAD1 came back as true positives, but the contaminating sequences, those came back as worm or fish. And so we ultimately pushed those out of our data set because they weren't true positives. All right. Okay. The transcriptome mining, it ultimately suggested that M. cerebralis contained NAD1 and NAD5 genes. These are the genes on the left and the e-values from the searches on the right. And we use an e-value benchmark of 1 times 10 to the negative 5. And generally, the lower the e-value is, the more significant and assured you are that it's a match. So with NAD1, pretty good. Still lower than our benchmark. NAD5, great. Really lower than our benchmark. COX2, which came back as positive, not good. Big no-no. Not lower than our benchmark. So ultimately, we just decided to throw these out because the e-value is too low. There might be too much divergence. So we can't make any assured statements on COX2 yet. This was a little bit of a side interest of mine, but we actually were able to obtain sequences relating to the NAD5 genome or the NAD5 gene from 2 separate TAMs. And that's what this graphic shows. On the upper x-axis, it is the sequence aligned from left to right, going all the way across from one organism. And then on the y-axis, same thing, top to bottom, it's that NAD5 gene aligned vertically. So if they were perfect matches, we would see a single straight line on the diagonal. That being said, we don't, which is pretty cool. What that could mean is there are translocations or genetic differences between the 2 organisms of the same species, but they maybe have a different type of gene or their genes are different. All right. So ultimately, the transcriptome mining suggests that M. cerebralis does not contain the COX1, COX2, or COB genes. Very clear. All right. But what does this, really mean? Where are we going from here? This is a picture of the electron transport chain, and the genes that we looked into, they're all involved in the mechanisms here. So we asked ourselves what function would be affected if those genes are missing. And what it kind of turns into is a pieced out subunit 4 in that. When you lose those genes and those genes no longer are present, you lose the proteins that they create. And ultimately, those proteins that, if they're no longer functional, you're creating a final quaternary complex of multiple proteins that are missing a function within it. So you can extrapolate from that and either say that you have an incomplete subunit 4 or a completely non-functional subunit 4. But that being said, the missing genes in the transcriptome, while they suggest they're absent, we need to verify that. And your transcriptome, it's just the expressed genes. If you're cold, your body expresses genes to make to make proteins that make you shiver to warm up. But it's really just a snapshot in time. So what we need to do is try and fill out those puzzle pieces and see what's actually in the genome, not just the snapshot in time transcriptome. So that's where our genome data comes in. With this, we're still working on it, but the steps that we outlined to continue our process is we're going to reassemble the genome. We're going to convert it to a searchable database. From there, we're going to blast those sequences. We're going to blast M. squamellus mitochondrial against that searchable database to see if there are any matches. Then we're going to map those hits to a reference assembly, circularize it if we can, so our beginning comes all the way back to our end. And finally, we're going to annotate those genes and try and identify what they are. And that's a look at our annotated circular mitochondrial genome. And that's what we want to get to. Just for some final takeaways, M. insidiosus, it does have a mitochondria. And we did not find significant evidence of COX1, COX2, or COB genes in M. cerebralis. That being said, where do we go from here? Someone's got to keep doing this research. I'm leaving, but we want to keep trying to figure out these problems. So this is probably where someone could go next. For continuing the investigation of M. insidiosus, that is going to occur at the genome extraction level. That's where I stepped out. That's where someone can step back in. There are a couple methods they could do. They could use the PacBio low and ultra low DNA input. So it would work for those low DNA inputs if we're not able to improve our extraction. There's also a circle to circle amplification, where you use the conserved genes within the mitochondria as primers. And then from there, you sequence based on that. Right. There's also, for M. insidiosus, we need to verify those results first, and then we can expand the search to other myxobolids. Verifying can be through additional sequencing and assembly, so we're just building that genetic repository of gene sequences and continually searching and making sure that our positives are positive. And then we can expand it. We can see if other myxobolids within either the cartilage, the muscle tissue, and other tissues are also losing their mitochondrial genes. Right. Ultimately, understanding the parasite energetics and what we've been doing, it can pave the way for future drug development and disease prevention. And really, it will help protect farmed and wild salmonids. All right. With that, thank you guys so much for listening and for being here. I'd like to open it up for any questions.